you so much. It's an honor to participate in this inaugural meeting and the consensus group. It's been really an exciting conversation. I've learned a lot about radiation and what we need to do, and we've talked about that. My role is to talk about the clinical evidence without radiation, and then the next talk we'll talk about radiation therapy and these same agents. So what's the current clinical evidence? My disclosures. So we know that antibody drug conjugates can enable tumor-specific targeting, although I have to say recent data has brought the tumor-specific targeting question into a larger question, because we know, and that this is important when we start thinking about the radiation therapy integration, is that we know that the targeting isn't so specific to the target now, based on our analyses that we'll briefly talk about. The whole idea with the new generation of antibody drug conjugates, or ADCs, is that they can have a presumed bystander effect. So the idea is that even in tumor cells that have very low, potentially unmeasurable expression of the target, you can actually have cidal activity because you have the drug incorporated into the higher expressing cells, which can be at a very low level, and then release of the drug into the neighboring tumor environment. And that's dependent on having a very high affinity antibody, so the delivery mechanism. The antibody itself has to have some activity, and that can be very difficult to differentiate. And then you have to have a highly active payload at very low concentrations because you're delivering small amounts. These are drugs that can't be given as naked drugs. And then the characteristics of the ADC are also very important. You have to have a linker that's plasma stable so that it doesn't release the drug into the circulation, and you have to have potentially a payload that is plasma or membrane permeable. So that would mean a relatively more water-soluble payload. Now, do you need that? There's actually ADCs now that have non-membrane permeable payloads that seem to be very effective. So that brings up some of this hypothesis into question. Also, the newer ADCs that are approved currently in the United States and in some countries in the world have a high drug-to-antibody ratio, but there are newer ADCs that appear highly effective that have a low drug-to-antibody ratio, but are utilizing this new technology of linkers and payloads. So it's actually a very interesting area right now where some of what we thought we understood, we may not be understanding quite as well as we thought. So there are a number of different ADCs either approved or in development for breast cancer. And what you can see here in pink are some of the agents that are farther ahead. The first two, trastuzumab deruxtecan or TDXD and sacituzumab govitecan or SG, are approved for a number of different subtypes of breast cancer. Datopotamab deruxtecan has completed phase 3 trial and ongoing phase 3 trials, and patritumab deruxtecan, a HER3 ADC called HER3-DXd, is still in process of finding a home in breast cancer. The disitamab vedotin will be in phase 3 trials and has had phase 3 studies in China, so we'll see more of that drug in the future. And ARX788 is being studied post-TDXD. And then there are a number of different ADCs, biphenotypic ADCs, et cetera, that are currently in earlier phase clinical trials. So antibody drug conjugates. These are a very exciting and effective drug delivery system. They appear to be effective across different subtypes of metastatic breast cancer. You know, we're talking about precision medicine. You know, we want to narrow it down and look at targets. My colleagues were just in a meeting last week that was focused on targets. Do you have a PIK3CA mutation or AKT or ESR1? These drugs seem to be effective across all of these different subtypes. So it kind of challenges what we're doing now, and we may want to deliver our chemotherapy primarily by using ADCs in the future as we continue to develop new ADCs with novel payloads. So we have an established role in HER2-positive disease, triple-negative breast cancer, HER2-low and hormone receptor-positive disease, and many ongoing trials in earlier lines, early-stage disease, and phase 3 trials. So we'll talk a little bit more about these at the moment. So DESTINY-Breast03 led to the final approval of trastuzumab deruxtecan, a trastuzumab biosimilar that's linked to a topoisomerase inhibitor called deruxtecan. Remember, none of these drugs can be given as naked drugs because they're too toxic at that dose. This drug had already shown tremendous efficacy in an umbrella phase 1 trial in HER2-positive disease. And one of my patients who was on that trial, DESTINY-Breast01, actually just went off drug at 5 years, having had just 1 year of disease control with trastuzumab emtansine. So this clearly is a huge addition to the armamentarium, even in those heavily pretreated patients. But DESTINY-Breast03 studied this in patients who just received 1 line, largely, of prior therapy for metastatic disease, median of 2 lines, 1 line with trastuzumab. About 2/3 of the patients received prior pertuzumab, and 15% had brain metastases. 50% of these patients had hormone receptor positive disease, as we see in HER2-positive breast cancer, that 50% hormone receptor positive and negative. What was remarkable about this trial, I mean, really incredible p-value with too many zeros to actually count. I think the initial data was p to the minus 27. So now they're just saying, less than a bunch of zeros and a 1. But this is the updated data that was presented at San Antonio last year and already published in Lancet Oncology. You can see that TDM1, the median PFS was 6.8 months, jumping to 28.8 months with TDXD. And this is one of the only trials we've had, maybe the only trial, where the overall survival was significant when the median hadn't been reached in either arm. So not reached in both arms. So then they did landmark analyses. And as you can see, at 24 months, 70% of patients were alive who received TDM1 and 77% who received TDXD. So really remarkable data. The other thing that was interesting was what people got after they received the ADC of trial choice. And you can see that only 17% of the patients receiving TDM1 received TDXD. So that was a question which then was answered by DB02, which I'll show you in just a moment. In the TDXD arm, 35% received TDM1, and that data will be very interesting in subset as we move forward and look at that analysis. In terms of toxicities, trastuzumab deruxtecan, and I think most important for this meeting, causes interstitial lung disease. And the rate overall, looking at multiple pooled studies, is about 15%, and it was in this trial too. This is the only trial with TDXD that showed no mortality from interstitial lung disease. The reason for that isn't clear, but it may be that prior treatment with trastuzumab reduces the mortality. The mortality rates are about 0.3%, as I'll show you in just a moment, but for whatever reason, there was no mortality in this particular trial. The most common toxicity from trastuzumab deruxtecan is nausea, something that we need to manage aggressively in these patients. Interestingly, this ADC and the other ADCs using deruxtecan as the payload don't cause a lot of bone marrow suppression. So you can have patients on this drug and they come out metastatic disease and they come in 3 months later and they're not anemic. We don't have any other treatment that we give that looks like this. So the other thing that's really interesting about these ADCs, we think of antibodies as being really big so they don't cross the blood-brain barrier. And this is also really important for our discussion and the next talk, which is that trastuzumab deruxtecan clearly has efficacy in patients with brain metastases and also in a small study in patients with leptomeningeal disease, both HER2 positive and HER2 low. Really fascinating data because we have essentially not much that works other than radiation for leptomeningeal disease. So it brings up the question of interaction between radiation and this antibody drug conjugate, where there's some data with TDM1 in brain radiation, as I'm sure you'll hear about next. So not only did they see a very good progression-free survival and overall survival in these patients with stable treated brain mets at baseline, but also saw intracranial responses. And again, in further studies, an LMD study presented at AACR last year, and also in a small trial called the DEBRA trial, there clearly have been sustained responses with brain metastases with this ADC. So DESTINY-Breast02 treated patients who'd already had progression of disease on TDM1. So this isn't the only trial that exists looking at an ADC after an ADC. In this trial, patients were randomized 2-to-1 to receive TDXD or treatment of physician choice. And as you can see at the bottom, this was sort of a after DB03, we already moved TDXD to the second line setting. So why does it really matter? Many people in the world are not going to have access to TDXD until later in lines of therapy. And it is a really important point about looking at an ADC after an ADC, because now this is a big area of study, particularly in HER2-negative disease. So as you can see, progression-free survival was dramatically improved with TDXD, going from 6.9 to almost 18 months. And remarkably, overall survival was also improved, 26.5 to 39 months, with a hazard ratio of 0.66. Again, ILD, and in this study, there was mortality, 0.5%. So in the analysis, pooled analysis of the risk factors for interstitial lung disease, more heavy pretreatment was one of the risk factors, as I'll show you in a moment. So that may have played a role in this study. So the approach to therapy for metastatic HER2-positive disease now incorporates this novel ADC, trastuzumab deruxtecan, known as TDXD to all of us. Tucatinib, trastuzumab, and capecitabine has activity in patients with active brain metastases, but as an oral regimen with some GI toxicity, generally in patients who don't have brain as their primary source of disease progression, this has been relegated to the third-line setting. And ongoing studies are looking at putting TDXD in the first-line setting, DESTINY-Breast09, that's looking at TDXD either with or without pertuzumab and comparing it to a Cleopatra treatment of THP. And that trial has completed accrual, so we're looking for that data in the near future. There's also a study going on in 500 patients who have brain mets or not at baseline. And then TDXD has already moved into the earlier stage setting, again bringing up a question for us about the safety of radiation therapy in DESTINY-Breast05, which is studying TDM1 versus TDXD. So TDM1, based on the Catherine trial, is our standard of care for patients with residual disease after neoadjuvant therapy for HER2-positive breast cancer. And radiation was given concurrently with TDM1 in the Catherine trial. There they reported a slight increase in radiation pneumonitis, 1.5 versus 0.7% in the initial publication, and there's been no long-term follow-up on those patients or what happened, small number, but still quite interesting, and no difference in radiation skin injury. So what happens in a drug that causes potentially fatal interstitial lung disease when you give radiation with the drug is a big question. This is a 1,600 patient ongoing study. And then there's a neoadjuvant therapy trial as well, DESTINY-Breast11. And that, I think, will be very interesting to look at what happens to those patients who've completed their TDXD, gone to surgery, and then have radiation post-surgery in a field that has to include at least some lung. So what about patients who don't have HER2-positive disease? The initial umbrella trial, the phase 1 trial, looked at patients who had low expression of HER2, a trial run by Daiichi, who initially was the only company marketing and studying this drug. So it was really interesting. Why would you study in patients who didn't have HER2-positive disease? TDM1 had actually been studied previously in patients with low expression of HER2, as was trastuzumab, and both of these investigations in the metastatic and early stage setting were negative. There was no benefit in those patients. But the idea here was with a better ADC, maybe you could actually have efficacy even if HER2 expression was quite low. And that started an entire new field, which previously we'd never really thought about, and now it's a huge area of investigation. So-called HER2 low disease, 1+ or 2+, without gene amplification. Now, I will say that this designation has been roundly criticized by our laboratory colleagues and pathologists who say that immunohistochemistry is totally inadequate for understanding low expression of HER2, and that really we need better tests that look at protein and gene expression in a much more sensitive way, and those studies are ongoing. But regardless, patients who had centrally confirmed HER2 low disease, hormone receptor positive, had a remarkable improvement in progression-free and overall survival in the DESTINY-Breast04 trial, leading to regulatory approval of trastuzumab deruxtecan in this population. In addition, an exploratory subset in patients who had triple-negative HER2-low disease also showed a remarkable improvement in PFS and OS, although this included only 58 patients with 18 in the control arm. So you would need to have much additional evaluation to really understand who these patients were who benefited, but we won't, because there are other ADCs with the same payload that will take this place in the future. So we'll see what happens. As you can see, response was also dramatically improved, 15.7 to 49%, and it was similar across subgroups, and HER2 low status could be determined in patients who had tumor from biopsies from 7 years before. And this is not the place to get into HER2 low determination, but there were many presentations at ASCO in San Antonio, a whole session on this area. As I mentioned, nausea is the most common toxicity, bone marrow suppression is uncommon, and interstitial lung disease was the most common reason for discontinuation. And 3 patients died of interstitial lung disease in this trial for 0.8% mortality rate and an overall rate of 12%. There's not a lot of cardiac toxicity, so that's encouraging in terms of the radiation overlap, but I think we really need to understand the risks with radiation and TDXD. This is a pooled analysis looking at risk factors. And, when we started thinking about looking at risk factors, we're going to be doing another pooled analysis looking at sequential therapy, et cetera, and retreatment. But, I said, what about radiation? This was a number of years ago with Destiny Breast 01. They were like, oh, we didn't track radiation. So it's an important thing. I mean, this is something we really need to be thinking about prospectively. You can see that the risks were age, being from Japan, interestingly. We've seen with other drugs having lung comorbidities, worse baseline renal function, a longer time since diagnosis, and hypoxia at initial presentation. And also, primarily, this occurs almost 90% in the first year after diagnosis, but mortality has been seen in a small number of patients many years after initial presentation. So it's something we really need to watch out for. We hold drug for grade 1 asymptomatic ILD with ground glass opacities on scans and stop drug permanently for grade 2 or greater. And that brings up another question of what do you do when there's minimal change based on somebody who's had radiation recently? Do you have to stop drug permanently? An ongoing study is looking at patients who have very low expression of HER2 called ultra low, so more than 0 by IHC, but not meeting the ASCO CAP criteria for 1+. 150 patients will be enrolled out of this 850 patient study, of course, have been enrolled since it's now completed accrual. There have been combinations with checkpoint inhibitors with TDXD, and in that, there was slightly higher rate of interstitial lung disease. So we do see more toxicity when we add a checkpoint inhibitor to TDXD. We'll talk about checkpoint inhibitors more tomorrow, but you can see that the efficacy in this first-line trial in triple-negative HER2-low disease was quite remarkable, looking at the waterfall plot. And then the spaghetti plot shows that the responses were very, very durable in this poor prognosis disease. So another ADC which is approved for the treatment of multiple subtypes of breast cancer is sacituzumab govitecan. This was a first-in-class TROP2-directed antibody drug conjugate that showed an improvement in progression-free and overall survival in heavily pretreated metastatic triple-negative breast cancer. And then recently we evaluated sacituzumab govitecan in patients who had hormone receptor positive but heavily pretreated disease where they had very limited additional options for therapy. These patients all had endocrine therapy, a CDK4/6 inhibitor, a taxane, and a median of 3 prior lines of chemotherapy for metastatic disease. And 95% of these patients had visceral metastases. What we showed was an improvement in progression-free survival, and it's interesting that we're still pretty crummy at actually differentiating who has disease sensitive to the treatment we want to give. You can see that by the first scan, 20% of patients had progression and fell off trial, but after that point, the curve separated and stayed separated over time with a hazard ratio of 0.66, and at 12 months, 3 times as many patients were progression-free who received SG compared to standard chemo. And then we showed an improvement in overall survival, and you can see this data here recently accepted in Lancet, actually yesterday. And what we saw here was that at 12 months, 47% of patients were alive who received chemo versus 61% who received sacituzumab. Now, we also looked at TROP2 expression. HER2 low, you get this HER2 ADC, very effective. And the same was true here. Very low TROP2 expression, we saw efficacy of SG. So there was no impact of TROP2 expression on efficacy of a TROP2 ADC, just showing you that we're not really good at looking at this particular target, HER2 or TROP2, with standard IHC approaches. And interestingly, we saw a high H-score in about 58%, but TROP2 expression was seen in almost all tumors. There are a number of ongoing trials here as well. First-line chemo and hormone receptor-positive disease, open and accruing, ASCENT-07. There are 2 trials, PD-L1 negative and PD-L1 positive triple-negative disease in the first-line setting. And the trial with pembrolizumab and sacituzumab govitecan out of Dana-Farber. There's a post-neoadjuvant trial through the German Breast Group, increased now to 1,320 patients and continuing to accrue, I think, near completion. And then the newly opened ASCENT-05 trial in patients with residual disease after neoadjuvant therapy for triple-negative breast cancer just enrolled its first patient last week under the direction of Sarah Taleni. The NCCN guidelines now list trastuzumab deruxtecan and sacituzumab as treatment options for patients with metastatic breast cancer across the different subtypes that we currently recognize by immunohistochemistry. Now, there is a new TROP2 ADC, datopotamab deruxtecan, or Dato-DXd. This was studied in the PanTumor01 trial, showing efficacy greater in triple-negative than hormone receptor-positive disease in patients who were fairly heavily pretreated, quite durable, and stomatitis, same toxin, deruxtecan. That we've seen the antibody before, right, with sacituzumab, TROP2, a little different antibody. Stomatitis, we haven't seen it with any other ADC. So really fascinating. We have no idea why. If you give the steroid mouthwash that we piloted with everolimus in the Swiss trial, you can markedly reduce the incidence and severity of stomatitis, but you don't eliminate it. So this will be quite interesting to follow over time. We have also studied Dato-DXd with durvalumab, showing significant efficacy, quite durable, again, stomatitis, and some pneumonitis when you give durvalumab from the checkpoint inhibitor. This drug does not cause pneumonitis as far as we can tell. TROPION-Breast01 studied Dato-DXd versus chemotherapy in second or third line therapy for hormone receptor-positive breast cancer. That trial has completed accrual. We're hoping for data later this year. And TROPION-Breast02 is an ongoing trial in the first-line triple-negative setting. And then there is a post-neoadjuvant trial called TROPION-Breast03, looking at Dato with or without durvalumab in patients with residual disease after neoadjuvant therapy for triple-negative breast cancer. And then lastly, the newest ADC with updated data, patritumab deruxtecan, also additional data presented at ASCO this year by Hamilton and colleagues from the Mini-Pearl network. Patritumab is a HER3 antibody drug conjugate, same toxin, though, deruxtecan. Its toxicity, again, is primarily nausea, but they do see thrombocytopenia, which we haven't seen with the other deruxtecan ADCs. So clearly the antibody makes a difference in what toxicity we see and may have a difference in interaction with radiation therapy. It appears to be quite active. I think it will be studied in hormone receptor-positive disease, and it has FDA fast-track designation for EGFR-mutated non-small cell lung cancer. We're actually studying Dato-DXd as neoadjuvant therapy in the I-SPY True neoadjuvant therapy. So it's our block A. People get 4 doses of Dato-DXd. And so far, anecdotally, we're seeing remarkable responses, and we're able to control the toxicity. So imagine if you could give patients 4 doses of Dato-DXd and go directly to surgery in some subtypes of breast cancer. And patritumab hopefully will be studied in that setting as well. So, very exciting and effective drug delivery system. I've shown you efficacy across the different subtypes. There are many trials ongoing, and toxicity management is clearly critical, and combination with radiation, which we'll hear about in the next talk, is largely lacking. A roadmap for metastatic triple-negative breast cancer and hormone receptor-positive disease now has to include antibody-drug conjugates, and I believe in the near future will include this in the treatment of early-stage disease as well. Thank you.